Hello, everyone. It's very exciting to be the final talk of this conference, especially because I present very novel work, and I'm very curious what you think about it. So basically, I will describe the new feature of high-C maps that we discovered in early embryogenesis. Basically, what I'm doing is studying major mechanisms of chromatin compaction and formation. So in our lab, Lignitz-Mirna lab, we study both of them. Loop extrusion, which is forming local features of high-C map, and compartmentalization, which is forming more off-diagonal patterns, far from diagonal, and reflecting the global chromatin organization. So the question is how both of these features of high-C maps, how they propagate to the actual function of the cell, and what do they change, and how do they affect the behavior of the cell. Of course, high-C matrix is just a representation of what's going on, and it's much more interesting to study the precise mechanisms. And we, several years ago, came up across this system where we have the rapid activation of the genome. So these are zebrafish embryos, and you can study different time points after fertilization. So somewhere here is 0 time point, where basically the sperm enters oocyte, and then you have the onset of zebrafish development. And then after that, you can probe different time points and trace what's going on with both expression and chromatin organization. And at around 3 hours post-fertilization, you have rapid activation of the genome. So before this time point, the embryo survives only on the RNAs from the maternal egg. But then, at 3 hours, you have rapid activation of its own genome, and it actually some very rapid and strong regulatory process, which is also very targeted to some specific genes in the genome. So what we did was take several time points before zygotic genome activation, shortly ZGA, and after. Apparently, if we take chromatin organization, there are no TADs at very early time points before ZGA, and they start to emerge somewhat around 11 hours post-fertilization. This is time point 5.3, and here you also do not see the TAD signature. And it looks like TADs appear only at and after 11 hours post-fertilization, which is pretty late after ZGA. So if we look at another feature of high-C maps at compartments, you will see that compartments appear only at 5 hours, and then it becomes stronger and stronger. But apparently, this is the global feature of high-C, but on a local scale, we see something else happening at these early time points after activation. And if we look at these features of high-C maps, we observe that there are some interesting patterns, weirdly shaped, and if we zoom in, they look like something that we call fountains. They look like enrichment of contacts perpendicular to the main diagonal. And this is this example of regular high-C matrix. This is example of observed over expected signal, where you can see this fountain signature much more clear. And if you take multiple locations of these fountains from the genome and try to average them, you will observe the structure like that. So actually, on average, pile-up of high-C matrices, you will also observe a nice average fountain. You can actually do this automatic calling of fountain patterns from your high-C data. We found 1,460 fountains across the whole genome of zebrafish. And this is how it looks like on average if we take the strongest peaks of fountain score. And then we can annotate these regions that we found as fountains. So actually, where these interesting structures emerge after zygotic genome activation. And it turns out that these are the regions of active chromatin. So there is enrichment of 27 acetylation, the chromatin is open there, and also there is binding of transcription factors, such as NANOC, which is pioneer transcription factor, one of the first transcription factors that are activating the genome. So it turns out that fountains are some structures related to regulation, and they are related to chromatin openness in the first place, and activity. But then we can do this interesting analysis when we take all the ataxic peaks, we annotate them by multiple histone modifications at this particular time of development of zebrafish, and then we infer chromatin states for these ataxic peaks. And we can infer 10 chromatin states, including transcription activity, enhancer, poised, repressed, quiescent chromatin states. And then we can try to annotate like what actually happens at the positions of fountains. And to do that, we can have some actual enrichment of this state at fountains, and we can have some randomized control. In gray here, we just randomized all the ataxic peaks, and we see that, yeah, we expect this distribution of enrichment to be located around 0.1. And we see that the red dot over here, actual position of the fountains, enrichment of the fountains, is located somewhat around this expected background. So actually there is no enrichment of promoters at our fountains. So the question is what's going on and what are the states that are enriched at the fountains? And it turns out that the only states that are enriched at fountains are enhancer related. It seems like only early enhancers but not promoters are forming these fountain structures. To prove it more, we can do it ab initio. We can just take whole the genome, and we can annotate it by, we can take ChIP-seq for trimethylation promoter mark and monomethylation of H3K4, which is enhancer mark. And we can split all the genomic beans into regions that are either enriched in promoter mark or enhancer mark. And then we can take aggregated pileups at this beans and take a look what's going on there in high C. And it turns out that only enhancers, an enhancer mark will be producing this nice fountain signature, and promoter mark will be instead forming insulation signature. So what else happens at these fountains? So this is like the strong support of the fact that fountains are enhancers, enhancers only. What else is going on at enhancers is actually if we take RED21 and look at the enrichment of RED21 across the genome, it looks like RED21 is broadly enriched at the positions of fountains. So what we would expect for TAB boundaries, for example, would be this very spiky enrichment of 21 because it stopped at the boundary of TAD, and there will be very broad and narrow, very sharp enrichment of RED21. But at fountains, instead, we see very broad enrichment, and this actually is coinciding with lack of CTCF. So if we take boundaries, there is an early signature of CTCF bound there, but at the fountains, apparently there is no CTCF. So our hypothesis is that these regions might be loading RED21, loading extruder, and that's why we see also some different pattern of RED21 around this loci. And the model that we implemented looks like that. So basically we have DNA strand, and in some region we have a particular loading platform for cohesin, and cohesin start load there and form this hairpin-like structure, very much similar to bacterial. For example, Bacillus subtilis. This same model will result in producing this diagonal perpendicular to the main one, but it will be very narrow and precise. Oh, and despite what we see in actual zebrafish fountains, is that the fountains are more broad. So they are not exactly perpendicular, they are broadening with the distance from the main diagonal. Yeah, just of note, I think it's not a secret that many people also discovered fountain-like structures in other biological systems, so it's not specific to bacteria, not specific to zebrafish, and probably it's much more broad phenomenon found also already in mice, for example, and in zebrafish sperm cells, for example. So if we try to simulate these fountains, we already know that, that we should avoid just narrow formation of the hairpin structure. We want to have something more broad and more shaped more like fountain and not hairpin. To achieve that, we can modify the model and allow introduction of background cohesins. So not only at the targeted site the cohesins will be loaded, but also somewhat in the background, and sometimes the collisions between cohesins will happen, and one of the arm of the cohesins will be stuck, and another one will be traveling. So that will create some desynchronization between arms of cohesin, and essentially this will result in the situation of broadening of fountains. So it won't be a hairpin anymore, it will be more broad structure because desynchronization of the legs of cohesin. And of course, we can propose multiple scenarios, other scenarios how this desynchronization can happen. So I presented you the simplest one when there is background cohesin. But also, it might be that there are some random obstacles on the way of cohesin. For example, it might be some CTCF binding which is happening at different locations, or it might be some weak boundary like MCM helicase, for example, which was mentioned before. Also, it might be due to internal properties of cohesin, when one arm is stepping independent of another one, and this might also create a nice broad fountain shape similar to actual Hi-C data. Interestingly, if we do the parameter sweep of the system, we can see that if we increase the background cohesin, so I showed you this nice example where there is reasonable cohesin background loading, but if we increase it furthermore, we will observe that the fountain is suffocating. So we remove the fountain by increasing the background loading of cohesin. And we also can do a modification of another parameter. We can just decrease the loading preference. So we can just say that there is very minuscule enrichment of loading at the target site. And this will also result to the disappearance of the fountain. And presumably that what we see in the development, because as we saw, fountains are only formed at this particular time point, 5.3, but then they can become any other structure, and they're not distinguishable in Hi-C maps, and they're not visible. And apparently it might be due to two reasons. Maybe there is raising of the background, so that we suffocate the fountain, or there might be the weakening of loading at target sites or at enhancers, so that we do not see any enrichment of the fountain signature. Okay, the next evidence for the fountains and that they are actually related to the regulation and might have some functional meaning, is that we performed knockouts of pioneering factors, one of them NANOC that I already mentioned, but alongside with NANOC there are two other which are very important, zebrafish, PO, and SOX. And we performed knockout of all of them in embryos, and we took attack-seek signal, and we created this nice plot. So basically on the x-axis, this is the signal of attack-seek, and here is the change in the mutant. So the regions that are located here, they were strong. There were strong attack-seek peaks in wild-type embryos, but they went down upon knockout of all of these three factors. And these regions in blue over here, these are strong attack-seek peaks, which are mostly unchanged. So they do not depend on the transcription factors. Now, if we create Hi-C pile-ups on these regions, we will observe that only those that were strong and went down, they were forming fountains. And those that were unchanged, apparently, they were not nice fountains in the first place. Then in the knockouts, we have a beautiful picture of disappearance of fountains that become just very, very weak. So it means that no attack-seek peak, no fountain. So probably it's one of the major determinants of the cohesin loading and the formation of fountain signature in Hi-C. The same goes with 27 acetylation. So we can do the same analysis for ChIP-seq of 27 acetylation in mutants versus wild types, and we also see that high down-regulated at 27 acetylation regions, they were forming very strong fountains, and they do not form them anymore in the triple factors knockouts. Another interesting thing that I really like in the system, that we actually can trace the destiny of the fountains in knockouts. And apparently most of them are down-regulated or unchanged, but some of them are up-regulated. So actually there are some compensatory fountains that will be formed at locations of the genome where there was some regulation happening, and in the mutant they will be trying to compensate probably for some regulatory troubles in the embryo. And now we have three different groups of fountains. We can try to see what were the factors associated with them. And apparently, transcription factors were mostly associated with those that are downregulated, so that are transcription factor dependent. And same goes with many other marks of activity, and that the functions that were downregulated there are supposed to be activated, and they have the strong activity signature. Interestingly, those that are going up, they do not have any clear signatures, right? Those that are upregulated. However, it turns out that these upregulated regions, they are future enhancers. They're enhancers that are supposed to be activated at 11 hours post-fertilization, but they do that earlier in triple mutants, and we can detect that by the positioning of fountains and seeing that the fountains persist in mutant embryos. So briefly, here is the story. And the summary is that we found the new structures, fountains. Their signature is that this is the signal which is perpendicular to the main diagonal of high C matrix, but it also broadens with the distance from the main diagonal. So it's not a simple hairpin. And we propose several mechanisms of how that signature can be formed. And the first requirement is that we need the targeted loading of cohesin. And second requirement is that we need some boundaries around, some random barriers around the fountain to create this signature, which might be background cohesin or some unknown factors, or maybe just decoupling of the arms of the cohesin. This targeted cohesin loading happens only at enhancers and not at promoters, surprisingly, and it also depends on pioneering transcription factors which activate the zebrafish genome. So this work was mostly in collaboration with Daria Nichuk from University of Freiburg, and Sergei Ulyanov performed data generation. These people were involved in bioinformatics analysis, and also Mirna Lab was involved in multiple discussions of this project, and I really appreciate this team. And finally, most of the tools that I develop here are with Open Chromosome Collective, Open to See, and these are also excellent people with whom I'm really lucky to interact and work. So with that, I think I will be super curious to learn your questions. Let me know, and I'm ready to take it. Yeah.
Questions?
Hi, wonderful work. So it appears that the properties that are predicted by these models of extrusion, they depend crucially on interaction between SMC complexes and barriers or other extruding proteins. And in a sense, in a lot of this simulation, we've always assumed that there's some sort of like static interaction, repulsive. But then we see Kes experiments, and it seems that cohesin can pass cohesin can pass almost anything else. So how do we reconcile these two things?
So your question consists of two parts. First is that you say that we assume extrusion model, which is local. Yeah, that's indeed true. There might be other models that are not assuming locality in this case, but we have some evidence that it might be not true. So it's not about just attachment of different distal loci, right?
No, sorry, this is not what I was asking.
This is the first part.
So
yeah.
The prediction of the model depends on how the different cohesin interact among each other and with other proteins.
Yes.
And we always assume some kind of repulsion or blocking because that's consistent with the high C.
Not necessarily. That's what I was discussing here, and that's the second part of your talk. Let's assume that it's extrusion indeed, and then do we actually need interaction of cohesin with some other obstacles on the way? Not necessarily. There might be decoupling due to internal properties of cohesin when one leg stepping is at faster pace and like randomly
That's also
switch.
ruled out by Kes-Decker. Let's say that it's substantially symmetric. So, I mean, Kes presented some result in which is saying that it's substantially symmetric stepping. So, I mean, like, I understand the difficulty, and I have the same. I just can't reconcile the single molecule experiments with the high C features.
We also should keep in mind that this is another species, so we probably we do not have the stepping properties of zebrafish cohesin. So that's why I'm a little bit hesitant here. But it also might be that these are indeed some random barriers on the way. Right? So we have multiple models that can explain it, which involve interactions with the background and surroundings.
But then again, Cass has showed many slides about the fact that cohesin can pass almost everything.
With some probability, but if you, for example, if you can pass the RNA polymerase with 80% chance, then if you have multiple of them, probably by just stepping on multiple of them, you will be stuck. So that's what might be happening here. Also CTCF, which might be just located not at precise boundaries, but some scattered around this area, around the fountain base. Maybe that can lead also to the same effect. But that's more weird scenario, because why would be CTCF not bound to one particular location, but to multiple regions in the area? So we don't know. Maybe there are some more factors that contribute to blocking of cohesin in zebrafish.
Hi, here. I'm sorry, but I have a very naive question. Do you think that this kind of fountain profile could be involved in the very high transcription, or it's quite independent?
Yeah, that's a very good question. Indeed, the genes that are located around the fountains, they are the first to be activated. And we see increase of transcription around fountains at this time point.
Hi, great talk. You presented these transcription factor knockouts. I was curious if you've also done knockouts of cohesin, WAPL, or NIPL, just to see how the fountain properties would change. Or I don't know also if there are mutants of cohesin that change its stepping properties to modulate like the angle or the intensity of the fountains. I'm just curious.
Yeah, thank you for the question. This is again zebrafish, and these are living embryos, so they do not like much interaction, much perturbations in their genomes, and without cohesin, they do not survive. So there is no simple opportunity to do cohesin knockouts in the system. And with WAPL and APBL, I don't think anyone tried that. So for that, I'm not so sure how to answer your question, but for cohesin, that is complicated, and that's why it's not done in zebrafish.
Hello, thank you for the talk. I'm curious. You mentioned transcription of the genes close to fountain. Did you look at the transcription of the enhancer? Could it be that gets transcribed also?
What I see is there is cage-seq available for zebrafish embryos at this stage, and what I see is that around fountains there is indeed active transcription, which can also happen at enhancers. So yes, there seems to be there is enhancer transcription, but apparently it does not result, which is surprising to me, it does not result in the formation of insulation pattern, because if we assume that transcription can interfere with the extruder, so it's supposed to create the boundary, but it does not create the boundary. It creates the fountain signature, which is very surprising. But the control, which is important here, of course, is what if we select the regions that have high signal of polymerase and low signal of polymerase around the fountain? Will the fountain be the same? And it turns out that it's the same. So it's independent of transcription in the surrounding regions.
Okay, I have one. Maybe I missed it because you said that the ataxic signal was the best predictor of fountain formation. Is it true for any ataxic signal or only the ones that are in enhancers?
Can you say it again?
The ataxic correlation, you say that it's the best predictor, the ataxic signal?
Oh, that's actually a very good question. We do not know what is the best predictor for the fountain signature, because there are at least 4 candidates, and we do not know in what sequence they step in the regulation of fountains. So first of all, these are transcription factor binding. like for example, like here, transcription factor binding, then formation of ataxic peak, acetylation mark, and also enhancer mark. And we don't know what of these four is the best predictor because we cannot have the system where one of them is perturbed. So when we remove just transcription factor, we see that all of them are gone and the fountains are also gone. But what is the best one, it's unclear for now.
Yeah, my question was, if you look at, I guess you have ataxic signal outside enhancers. So here you are aligning only the ones that are, so maybe it can help to say that ataxic, when they are not in enhancers, probably they don't form any fountain.
Yeah, there are some ataxic peaks that are not enhancers. There are some ataxic peaks that are not promoters and not enhancers, and apparently they don't form fountains. Okay. Yeah. So also a good control, for sure. Yeah.
Aleksandra and all the speakers
Thank
of
you
this
for the
session.
questions.